Good morning to all, and I request Dr. Seema to kindly time me up. And today I'll be presenting on an interesting aspect of circadian rhythm and the liver clock and possible implications in normal physiology and in disease. But before I start this presentation, I would and tell about the outline what I'm going to speak. I'll start with why the study is important, and I've picked up a paper from Dr. Manoj's work where they have done the sleep-wake abnormalities in patients with cirrhosis. And what this paper suggests is that 65% of the cirrhotics have the sleep-wake abnormalities, thereby indicating that there is a circadian misalignment. And accordingly in the paper, and Dr. Manoj can tell better, they have put the patients into various chronotypes, evenings or the morning types. And this paper has been very nicely cited up by a recent paper in Lancet, Gastroenterology, Hepatology, that talks about the liver diseases with respect to the sleep disorders. And what is best about the study done by Dr. Manoj is that of all the papers that they have reviewed, the table runs in 2 pages, and from 1999 to 2017, the sample size is 44 to 130. And post that, if you see, the study done at ILBS has the maximum number of subjects enrolled. So this indicates the importance of the sleep disorders, and for that matter, again from the same review article, whatever be the disease etiology or the background, it is involved with disturbances in the thermoregulation, again linked with the circadian or the biological rhythm, and circadian misalignment and blunted responses in the disease. Thus the rationale for picking up this topic. So this is the outline. I'll first start with the definitions. Where exactly, so when you talk about this rhythm, you need to know where the clock is, what all the types of the clocks, what are the components, whether it is clinically relevant or not, and what is so special about the liver. And finally, if I get time, I'll just briefly discuss some basic biology questions that we tried to address. So coming to the definition, the biological clock is an internal physiological rhythm which synchronizes with the outside environment. Most of the time, the outside environment is perceived by the light and the dark. However, there are other activities also that helps in the synchronization, especially the food and the physical activity. And when you say circadian rhythm, it is usually, the time is a cycle of 24 hours, where you have most of the work which I'll be discussing is with respect to the mice, so with the rodents, so I'll talk in terms of that, where you have a 12-hour night and a 12-hour dark cycle. There can also be infraradians, that is the rhythm less than 24 hours, or ultraradians where it can be more than that. So the clinical relevance, the clinicians know it much better, be it you take the temperature or the blood pressure, they all cycle throughout the day. And the best example, of course, as a clinician, you know it much better than me, and this is the cycling of the cortisol early in the morning, which is the Brahma Muhurat so-called, and the picking up of the melatonin in the night. So this is what the rhythm is, and almost all the images that you will see in consequent size, they will have this typical rhythmic pattern where you have an amplitude, where you have, so the amplitude that's called the nadir or the depth, and then you have how the rhythm is in a 24 hours, 48 hours, or a 72 hours. So now the next question is, what determines this rhythm? The rhythm is determined by the clock, and basically there are 2 clocks. One which is present in the central nervous system, which is the central clock in the brain, and then you have the peripheral clocks. Now the central clock is response to the wake and so the activities. So with the light through the retinohypothalamic tract, the signals reaches the brain, and in the brain there are these special neurons in the suprachiasmatic nuclei. This suprachiasmatic nuclei through the brain then talks to all the peripheral organs. So that means the brain and the peripheral organs have to be in synchrony. Not only that, the peripheral organs in the others like the kidney, liver, lung, etc., they all also have to synchronize amongst themselves so that they don't go haywire. All this is determined by a complex molecular machinery, which I'll be discussing in the next slide. So what is the pacemaker? So I told Dr. Seema ma'am to actually time me up. So here the timing is, from the SCN, the timing which is given to the different periphery is again through the hypothalamic pituitary adrenal axis and the glucocorticoids. I'm not going into the details of this because I myself don't know much about it. The sympathetic tones, and that's why I think neurohepatology is important. And not only that, it's the physical activity, the thermoregulation, and more importantly, the meal. So it's not what you eat, but also what is the time of the eating. All these determines the synchrony or the rhythms, that is which of these genes are going to be expressed, when they are going to be shut down. And this was recently reviewed in an article, and this is in praise of the liver, where besides the central clock and the periphery, among the various organs, liver has been given a higher hierarchy. The reason being, if you delete any of the clock in the liver, the two organs which get affected, at least in the rodents, one is the lung, the other is the adipose tissue. So that tells about the importance of not only the central clock, but also the peripheral clock, especially with respect to the liver. Now that's okay at the level of the organism. So you have the clocks at the level of the organism. It's evolutionary conserved from small organism like a Drosophila to a higher mammal. It's there in all the organs, especially in the higher organisms. It's also ingrained very nicely in each of the cell type, and it is ubiquitous. So what is the molecular components to it? The molecular components are the two transcriptional factor, clock and the BML. The clock stands for Circadian Locomotor Output Cycle Scapula. So it's a big name which was first discovered actually in the Drosophila, and then later on the homologs in mouse as well as the mammalian homologs were found. All the work done in the Drosophila actually ended up giving a Nobel Prize for Hall, Robash and Young, because they actually very nicely, using the genetic mutants, dissected the machinery. So what is this machinery? During the daytime, there are two transcription factor, clock and BML. They work as a dimer. In the night, the dimer is cry and purr. So BML stands for brain and muscle protein, and period is the purr, and cryptochromes is the cry. So the positive feedback is through the clock and the BML during the time, in the daytime, when the BML goes up. They both heterodimerize, and what they do is they go and bind to the promoter regions of certain genes, of which the purr and cry, again the clock components are there. Besides that, there are a lot of these CCGs, which are core clock control genes, and this includes genes such as those involved in glucose metabolism, insulin secretion, lipid metabolism, bile synthesis. So you can imagine how many genes these two molecules control. So when they build up during the morning time, that is the active time, at least from the human perspective, and in the night time, activity for the rodents. They do a lot of transcriptional processes, and in the night time, when the activity is low for the humans, and in the morning time for the mice, the cry and purr, what they do is they go and block the clock and the BML. So this is called as a transcriptional translation feedback, because this is a clock which goes around just with these four basic molecules. And what these outputs are? The outputs is what is the clinical relevance to all of us. The sleep-wake cycle, the hormonal oscillation, body temperatures, cell cycle division, and most importantly, the metabolism. So I'll just pick up this metabolism part and explain. So again, recapitulating, the human versus the mice difference. The BML and the clock are high up during the activity time, and during the sleep time, it's the cry and the purr which comes into the picture. So there is a circadian rhythm of any gene. So whatever genes they are expressing, the clock and BML, they will go up during the morning time. Then during the night time, when cry and purr comes up, the genes is shut off, so you have a trough going down, and then again in the morning time. So you have, this is what is the biological rhythm. And the liver senses it through not only the SCN, but the light, meal, and the activity time. And the genes which are controlled in the liver clock are those with the nutrition. So you have the AMPK and the mTOR, which is very important for the metabolism, the autophagy, most importantly the glucose homeostasis genes involved, the lipid homeostasis, especially the cholesterol, and cholesterol again connects with the bile, and in the organelle level, both the ER, which is the endoplasmic reticulum, and the mitochondria also show a rhythm. And this is a typical rhythm which is actually done using the mice, where this white box indicates the light. So time point 0 is when you turn on the light. So in our animal facility, by 6:37, the lights are turned on. And then by 6 or 7, they are actually shut off. So you have a 12-hour light period and a 12-hour dark period, which is given by the term Zitterberg. And there is a cyclic change in the gene expression. Now this red line here indicates the mice which has been given a high-fat diet. And you can see the rhythm is totally disturbed out. So very briefly, liver is a pulsatile organ, and you have almost everything in that has a circadian rhythm. Mostly this work started with the gene expression, and with the omics technology, now you know that proteins are rhythmic, lipids are, metabolites are, and so on and so forth. So this is the classic paper which talked about for the first time the liver clock and how the liver clock is involved with the metabolism. So here what they did was they took the animals for almost 24 to up to 48 hours and sacrificed the animals at every 4-hour interval, and then they did extensive genomic study with respect to the mRNA transcription. And what they found was the genes which are gluconeogenesis, glycolysis, fatty acid, they all peak at the activity time. Since it's a rodent, it's actually peaking at the night. And then again there is a trough, as I was saying, and then again there is a pickup. So all these genes which are in the blue are showing a cyclic nature. So this, and also what they found was the genes which are involved in the glucose transport, especially in the intestinal, the GLUT1 and the GLUT5 and the GLUT2 at the in the liver. So they did it in all the organs, and they found that even these transporters have a cyclic rhythm. So this is again an expression recapitulating BML going up in the night time, at least for the mice. And when it is going up, it is then causing a cyclic change. And the earlier one was with the glycolytic pathway and the oxidative phosphorylation. This slide here indicates that both cholesterol and bile metabolism also show a circadian rhythm. So they found that the enzymes which are of cholesterol biosynthesis peaks up at night. Mind you, again, this is in animals, so you have the other way around in the humans. Then the enzymes which are helping in the cholesterol degradation, which then leads to the bile acid production, they are also regulated. So these indicate that important lipid metabolism and glucose metabolisms have a circadian pattern as seen in the rodent studies. Now this is with respect to one organ, but what about synchronizing all the organs? So brain is now talking to the liver, but how then liver is talking to the rest of the organs is also important. So this is one good example taken from American Journal of Physiology, again done in rodents. So the gallbladder gets filled, this you all know, and then the bile gets released out whenever there is an activity and the food is taken. Now this is concomitant with the changes in the bile profile in the intestine. Then of course it's circulated back through the portal circulation into the liver, and then also it's there in the serum. So you can see that there is very nice synchrony of clockwise phase and an anti-phase with respect to the gallbladder. And all these is connected because the genes which help in the transport of the bile, like NTCP, BSEP, they all have a circadian rhythm. So this makes it important that maybe cholestasis, where there's an accumulation of the bile leading to injury, could there be a disturbances in the expression as well as the rhythmic profile. So cutting the story short, you have a normal biological rhythm, and when you have something like a high-fat diet or an alcohol, the rhythm gets displayed. And this is taken from a review article which very nicely says what all are the pathways upregulated in the morning times and what are downregulated during night times, and you as a clinician know it much better. And whenever there is a circadian misalignment, either with high-fat diet, so the whole concept, with at least with the mice came, there's a concept of jet lag. So you are either moving eastward or you are moving westward. So either you gain the time or you lose the time, especially in the travelers. But if you are a couch potato, you don't even have to travel. Just the food and no physical activity can actually totally misalign. And that terminology is now being given as a social jet lag. So we recently compiled up all the literature, and we find that if these genes are, especially these 4 genes of BML, CLOCK, if they are deleted in the mice model, what is with respect to its fatty liver phenotype? And these are all the different targets. I'm not going into the details. So Basically, the underlying story is that there is dyslipidemia, there is insulin resistance, and leading to steatosis more. And when you overexpress, as has been done in one of the study, when you reconstitute back the BML, the rhythm is turned down. There's another way that you can turn on this rhythm. So the keto diet can actually put back the rhythm back. That's what the literature says. And also, if you just do a time restriction, that can also put the clock to the right time. So I didn't go into all the details on alcohol and the fatty liver because of shortage of time, but just quickly 2 or 3 slides on what is special about the liver clock. So there are 2 things which are important. One is its autonomy and its independence. So this is in praise of the liver. So when there is, so this is again work done in mice, published in Cell, where the liver is in synchrony with the brain. So the question is the central clock the only master orchestra that composes all the music? Not so. The liver can do it on its own also, independent of the brain. So in this experiment, what they did was they had a simple mice, which was a wild type, and in another mice, they selectively deleted out in 2 ways the suprachiasmatic nuclei, and also in one experiment, without damaging the nuclei, the nerve cell, they just shut down the expression of the core clock components, BML and the clock in the brain. And then asked the question, what happens to the rhythm in the liver? When they do this, they found, so let's say the first scenario, the wild type, you have almost 100% of the transcripts show a pulsatile rhythmic fashion, especially those in lipid metabolism and the glucose metabolism. When you remove the SCN or damage the clock in the SCN, then the BML is still, the periphery clock is still there. You lose most of the rhythm, but some 30% of the transcript, they are maintaining the rhythm, independent of the brain. And what are these? These are those related with the glycogen, glucose, the NAD, which is the energy metabolism, and the redox. So these are the basic principal components which actually helps in the survival. And that's maintained by the liver on its own as an autonomous. However, for this cue, what the liver needs is the light. So you can think that liver is an internal organ, so how is it going to see the light? So what these people did was they kept the mice in a totally dark environment. Again, there is no SCN. I'm sorry, the cross is gone here. The SCN is gone, but the BML is still working in the liver. But if this is exposed to a total constant darkness, this rhythm gets disturbed. So this work indicated that liver can detect changes in the environmental light through certain mechanism which we still do not know, independent of the brain. And brain, at least, though it is a master clock, but some of the peripheral organs can do in an autonomous way maintain their own rhythm for a homeostasis and survival. Now this is another interesting paper. So that was with respect to the metabolism. Now what happens to the liver size and the liver volume? Now in this experiment, what they did was, if I can enlarge it up, they found that the liver oscillates during the day and the night time. And this is a image of the liver and the hepatocytes. And what they find is that the hepatocytes, to begin with, are maximum in size at the time point 0. So time point 0, ZT0 is the time when the mice is waking up. And sorry, the lights are turned on. So the activity is now going to go down. And when it is actually in the sleep mode, the liver is actually shrinking. But then again, in the night, the liver comes back to its shape because now this mice is actually active. So just looking at the morphology, that is both the weight. So what they find is that there is a circadian rhythm with respect to not only the size of the hepatocytes, but also when you put that in perspective with the liver to the body mass, again you see this fluctuation. And what they find is that this fluctuation is only there with the liver, but not with the rest of the organs. So this indicates again its hierarchy in the clock. However, if you shift the feeding time so that the mice is actually a nocturnal, so in the morning it is sleeping. But if you make a rhythm that you don't let it sleep and you put it for a feeding, and then don't feed it in the night time when it has to be active, all this rhythm gets disturbed. So this indicates the importance of the liver volume and the weight with respect to not only the feed, but also the time of the meal, also as its activity. So this is what it is. At the zenith, when the animal is the most active and now it's going off to sleep, when the lights are turned down, the liver mass is the maximum, and again in the night when the activity goes on, it slowly regenerates. So it's like how it was in the Greek mythology of the liver regenerating in the night. And all this has been now linked with the ribosomal biogenesis at the molecular level, which I'm not going to go into details. So this is fine with the rodents. What about the liver? And then when I dug the literature, there's only actually, maybe there are other papers at least, but I found that there is only one paper where the volume of the liver has been measured by the ultrasonography in normal subjects and alcohol. This is a paper published in JHAP in, I think it was JHAP in 1986, and they have just taken 8 subjects. And what they did was, from early morning 9 o'clock to 4 o'clock, they have actually measured the volume. And what they find is, they did 8 subjects on 3 consecutive days, and what they claim is that they have a very reproducible result, that between 12 to 1 in the noon time, that's when the maximum depth of the liver takes place in a normal subject. The liver volumes go down. I don't know, as a clinician, you noted this or not. However, in the alcoholic, this diurnal variation is not there. The other limitation was they did not follow it up throughout. I know it's difficult to wake them all up every 2 hours to check this thing, but this is an interesting concept that the liver volume is also. So unlike the other organs I do not know about, but liver is one thing where the volumes is also. So it's autonomous, it's also has a fluctuation with respect to the volume. Now, many of you are interested with regeneration, so this is another, again, done in rodents. So what happens to the liver regeneration with partial hepatectomy? If you do it in the morning time, or you do it in the night time. Though there are limitations in the study, but what this paper suggests
is you have another three minutes to go.
Okay. So the night retards. So if you do a partial hepatectomy at the night, there is a long time for the liver to regenerate. And how did they do this? They have done it through the Ki67 staining, which comes up much more when the liver is dissected in the morning time. How much of these is applicable to a clinical scenario where the surgery is done in morning time versus night time remains to be seen, although there are limitations. But we do find that when you give the mice the high-fat diet, or if you just put increased amount of palmitate, as we've done with the cell cycle analysis, there is definitely a delay in the cell cycle phase. So that means the times are all disturbed out. This leads to another important topic, which I'll just very superficially mention. This is chronoimmunology. So this is how the stem cells come into the circulation during the rest time, and then again they go back during the activity time. So that means the regeneration process is much more active during the night. So if I have time, I'll quickly go through some of the work we've done, and once I'll sum up the work which was done by Savera, where we've looked at the clock in relation to the sirtuins, and the mediator for this is a metabolite, which is the NAD. So what we found is that palmitate, when you feed it to the cells, it leads to inflammation. And it leads to inflammation, it is also causing cytotoxicity. So we asked the question, can we work out the mechanism by which we can actually stop this? And what she found was that when the palmitate is given, there is a disturbance, at least in the clock. So this green line indicates the circadian rhythm of the clock genes, and this is that of the BML. So we were expecting even the BML will go down, but you see the BML, there's only a shift, and there's an acute peak, and then there is a shift. So BML is disturbed, but it is only by 4 hours of phase. What about the sirtuins? The SIRT2 follows a pattern similar to the BML. So we asked the question whether BML and SIRT2 are synchronized or not. And indeed the answer came yes, because the BML during the high-fat diet went down, and what we found is that the SIRT2 promoter has a site onto which the BML binds. So what is the mechanism through this? What we found is that the palmitate actually inhibits a rate-limiting enzyme, NAMPT, which helps in the making of the NAD. So if the NAD levels are fluctuating, the SIRT2 doesn't work because NAD works as not only in the oxidative phosphorylation in the mitochondria, but also as a cofactor for the SIRT2. So basically what we are saying that either you pump up the SIRT2, so even if the rhythm is disturbed in a high-fat diet, if you can somehow pump up the SIRT2, and we've done by overexpressing it in the cells, we can actually delay out the cell death and also prevent to an extent the lipoinflammation. But more importantly, this is not feasible all the time. So what we found is that NAD, you cannot directly supplement it, but NAD, one of the precursor is the vitamin B3. And vitamin B3, the bioavailability is low, so we picked on to the nicotinamide riboside, whose bioavailability for NAD is more. So when you give the NR, actually these processes are inhibited out. And with respect to the clinical samples, we could not do a serial this thing, but what we found that BML, at least in the NASH, and this is thanks to Dr. Archana, we could do, it's very nicely present in the nuclei. But in the later stages of the NASH, where it is with the cirrhosis, everything comes out. So this kind of indicates that there is a disturbance in the protein expression, which could be leading to a misalignment. And again, when we did the metabolomics with Dr. Jassi, we found that in the NASH, we had more of the NAD, surprisingly, but in the cirrhotics, it was actually a dip. Now, it could be more NAD circulating in the plasma because more cells are dying because our CK18 was also high up, or maybe it's kind of a mechanism forwarding out as a feedback loop, which we do not know yet. Now, this is an ongoing work. Very briefly, I'll tell you, this is, we try to make a social jet lag mice together with Dr. Anupama. And what we are doing here is, this jet lag mice, we are, for every week, twice in a day, we just turn on the lights early. So you have 12 hours light and 12-hour dark cycle. But now these mice see less of the light and more of the dark. So in this, we again put them into 4 groups. Those with regular diet, high fructose and high fat, which is equivalent to social jet lag. And we supplemented it with the NAD and asked a few questions. So this is a preliminary data. We find that these mice have high fasting glucose, maybe indicator, but more interestingly is their phenotype with respect to the activity. You see, this is a normal mice, which works very nicely on a wheel. And this is a jet lag mice, a little confused, poor chap, and it's trying to do it. But then if you keep on making them exercise through the wheel, it actually picks up. This is still, again, as I said, pilot. We've done a micro CT, and we find that these jet lag mice have low, high abdominal girth, and also it has high abdominal fat. This is the, I'm sorry about this, this is the HND staining, which is showing that the jet lag has more steatosis with inflammation, which is not so much in the control. And also they have fibrosis in both the groups. And we thought that NAD will work in both the control arm as well as the jet lag arm. And intriguingly, the NAD works, reduces the fibrosis only in the jet lag group, but not so in the control. We do not know why, but we still have to do a lot of more scoring with Dr. Archana now. So coming to the end, this is a snapshot which I picked from Journal of Hepatology, the recent one, which basically sums up the pacemakers, which controls this rhythm, the feed, the activity, and the light. And this is at level of protein, lipid, metabolite, everything, and even the cell size and the volume. And you have the process, at least in the mice system, during the night time, the regenerative process is high up. So in summary, the liver clock oscillation is linked to the metabolism. What you eat matters, the timing matters, and it is important to have a rhythm. And there is now new concepts which are coming on, chrono nutrition and chronotherapy. That is, the time of the drug needs to be given when the BML is actually down. Maybe can you pump that BML up or not? I didn't go into this. And our own data, we are showing that SIRT2 is regulated by the BML. And finally, all the departments I thank, because extensively we worked with them. And much of the work I showed with Savera, we are still writing up the manuscript. And Dr. Anupma, who's doing the ongoing work on the jet lag. Thank you.